Cell metabolism, part 1. What is metabolism? Metabolism is the creation of energy and building blocks for the cell. It's really all of the chemical reactions that take place in a cell. Making energy in the cell, we know that chemical bonds hold energy, and making and breaking bonds takes energy. Cells and organisms take in energy in the nutrients that they ingest, but the chemical bonds holding the energy in those nutrients are too spread out for the cells to use efficiently. So the energy from the chemical bonds in the nutrients have to get compiled together and transferred and concentrated into ATP molecules. And the ATP can then release that energy quickly to power cell reactions. There are two kinds of cell metabolism, catabolism and anabolism. Catabolism means breaking things down, and anabolism means building things up. When you are breaking things down, you release energy, and that energy can be used to make ATP. This usually happens through hydrolysis reactions. In anabolism, it's the opposite. You're putting things together to make bigger molecules, and this requires a great deal of energy input that usually comes from ATP. These reactions usually involve dehydration synthesis reactions. Remember those? We've talked about them a little bit before. They release water. Here's what the catabolism-anabolism cycle looks like in our cells, right? We have glucose that we take in as energy. That gets broken down. ATP is made, and that ATP is then used to put building blocks together through anabolism to make things like proteins. The most important kind of reactions that take place for cell respiration are oxidation reduction reactions, right? Because the energy in the nutrient molecules is associated with electrons. As nutrients are broken down by catabolism, the energy is transferred to generate ATP. Those electrons have to get transferred through redox reactions, also known as oxidation reduction reactions. What those are there are two coupled reactions that always take place together. One molecule loses an electron and the other molecule gains an electron. When a molecule loses electrons, we call that oxidation. When a molecule gains electrons, we call that reduced, hence the term oxidation reduction or redox. Okay, here's what that looks like. You have molecule A that starts with an electron. It loses it and gives it to molecule B. Since molecule A lost an electron, it's said to be oxidized. Since molecule B gained an electron, it's said to be reduced. There's a really great acronym that I learned in college that helps you remember which way the electrons are going and which word is which. It's LEO goes GER. Lose electron oxidized, gain electrons reduced. Leo the lion goes GER. When those electrons are moved, protons are also moved at the same time. When you have an electron and a proton put together, you have a hydrogen atom. So here's a great example of moving an electron and a proton at the same time. Okay? When we have an organic molecule like glucose that is oxidized, okay, while it's being broken down, meaning it loses an electron, you have something like NAD, which gets reduced and gains an electron. Its chemical formula goes from NAD to NADH. That H is an entire hydrogen atom, but it is an electron and the proton that goes with it. Okay. Why are redox reactions important? This is how ATP gets made in our cells. When we're making ATP, we have to put in a lot of energy. The bonds that hold that third phosphate onto the ADP are very unstable and they're easily broken. It's like trying to force two magnets together that don't go together, right? And the opposite of that, when ATP gets hydrolyzed into ADP and PI, there's that tremendous release of energy. When ADP and a phosphate group make ATP, we call that phosphorylation, okay? What we're going to focus on is the cellular respiration of glucose, or breaking down glucose to make energy. This is the most commonly studied form of cell respiration in biology, but there are lots of different sugars that can get broken down, but they all kind of happen the same way. The big thing is that when all of these bonds in this 6 carbon, 12 hydrogen, 6 oxygen molecule get broken down, energy is released. Okay? Energy is released slowly in this case and put into ATP in our cells. Okay? When energy is released quickly, you get a rapid release of heat and light, which is fire. Okay? So what we're talking about here is carbohydrate catabolism, the breakdown of sugar, right, to make energy. So overall carbohydrate catabolism could go one of two ways. It could go through the cell respiration pathway, or it could go through fermentation. We'll talk about both of them, but we're first going to talk about cell respiration. Cell respiration can only happen if oxygen is present. Okay? Respiration consists of glycolysis, the Krebs cycle, and the electron transport chain. What it is essentially is a group of redox reactions that take all of the electrons from glucose and give them to an enzyme called ATP synthase, who then makes ATP. Okay. There are some really important players we have to talk about in cell respiration, and they are the electron carriers. Their names are NAD, NADP, and FAD. These molecules will become reduced, meaning they gain an electron, and their names will end up with an H at the end, as those glucose pieces are broken down and oxidized. These electron carriers, once they are reduced, will then travel to the electron transport chain to help power the making of that ATP. The first step in the pathway is glycolysis. Glycolysis will actually happen no matter whether you go down the cell respiration route or the fermentation route. You're essentially taking glucose and oxidizing it, breaking it down into 2 pyruvic acid molecules. This also produces a little bit of ATP and a little bit of NADH. All right, so in glycolysis, we have our glucose being broken down into 2 pyruvate molecules. We gain a little bit of ATP and a couple of electron carriers that have gained electrons and become reduced. All right, whether you are a prokaryote or a eukaryote, this process occurs in the cytosol and it does not require oxygen. Okay, so whether you go on to the rest of cellular respiration or you go on to fermentation, this process of glycolysis always happens. What it is in more detail are 10 enzyme catalyzed reactions, each reaction with a different enzyme, different substrates and products. Most cells will do glycolysis, so we think it evolved very early. Okay, it uses 2 ATP, but you gain 4 for a total overall net gain of 2 ATP. Okay. Here is what those steps are, but you don't need to know this. This is just here for your information. Okay. After glycolysis, we have that pyruvic acid, right? Or pyruvate, same thing. It has to get modified before it goes into the Krebs cycle, which is the next step of glycolysis. And in that modification, it gets turned into acetyl-CoA. And you have another electron carrier that gets an electron, and you have a little bit of carbon dioxide coming off. Okay. For most purposes and for counting ATP, this is counted in as part of the Krebs cycle. The Krebs cycle itself is literally a round cycle because the enzymes and molecules get reused as the cycle goes around. And what it does is it takes those 2 molecules of acetyl-CoA, which come from that one glucose nutrient that was originally ingested, and it breaks it down over the course of the Krebs cycle and removes all of the carbons and breaks all of those chemical bonds. And this is where all of the electron carriers get reduced and we get a lot of electrons out. Okay, so essentially this acetyl-CoA gets turned into citrate, which is why this is also called the citric acid cycle. We get a little bit of ATP, but we get 6 NADHs and 2 FADH2 molecules. Now these are both our electron carriers. In eukaryotes, this happens in the mitochondria, but prokaryotes don't have mitochondria, so this happens in the cytosol. Here's what the Krebs cycle really looks like if you want to know. Again, way too much biochemistry in detail for microbiology class. All right, the last step of cell respiration is the electron transport chain. And this is where we have all of those reduced electron carriers that now have those electrons. They go here and they deposit essentially their electrons into the electron transport chain. And the electrons flow through a series of 3 enzymes where the electrons get separated from their proton in the hydrogen atom. Okay. And the final 4th enzyme in the series is ATP synthase, and that is the one that makes all of the ATP. All right. So the first 3 enzymes in the electron transport chain, they are going to separate the protons and electrons from the hydrogen atoms. They're going to pump the protons across the membrane, right? And the electrons will go down the chain, eventually making their way to oxygen molecules to make water, which is why oxygen is required for cell respiration. Another key thing for cell respiration is after those electron carriers give up their electrons, they get recycled and they go back to glycolysis or the Krebs cycle to pick up more. So we reuse them over and over again in our cells. So something important to note about the electron transport chain is its location. In eukaryotes, this happens inside the mitochondria on the inner mitochondrial membrane. But prokaryotes, remember, don't have mitochondria, so this actually happens on the cell's plasma membrane because it's really the only membrane the cell has. Here's a really great picture of what the electron transport chain really looks like. So you have these 4 enzymes, these big proteins in the membrane. Right, the 4th enzyme is ATP synthase. The middle are things like NADH dehydrogenase and some cytochromes. Those are kind of more minor. The big player here is ATP synthesis, but all of these 3 have very important roles as well. Okay, so you have the electrons from your electron carriers. They're coming to the electron transport chain. These enzymes are taking those electrons and protons off from the electron carriers. They're pumping the protons across the membrane and transferring the electrons down the chain where they eventually get combined back with their protons to make water. So here's a great picture showing the flow of the hydrogens. So you have your NADH, your electron carrier down here that comes in, right? You take off the hydrogen and you get NAD plus, which goes back to other places in cell respiration. Okay, you send these electrons through to the other enzymes in the chain, and you pump the hydrogens across the membrane. This creates a buildup of hydrogen ions, and they're all positively charged, and like charged molecules repel one another. So they really don't want to be around one another. So as this concentration builds up, they want to be around each other even less. And so this creates this really high energy gradient that occurs because these hydrogen molecules, after they build up, they really want to get away from each other. And to get away from each other, they flow down through this ATP synthase molecule with so much energy that it powers the synthesis of ATP. All right. So electron carriers, they split that hydrogen into an electron and a proton. protons get pumped across the membrane, the electrons get passed down the chain. Okay, the protons will form a gradient, go down through ATP synthase to power ATP. Once the protons have come back down through ATP synthase, they recombine with their electrons as hydrogen atoms, and they combine with oxygen, making water. Another picture, same thing. All right, so let's summarize respiration. ATP gets produced from the complete oxidation of one glucose molecule. This means we're taking that one glucose molecule that the cell originally ingested, we're breaking down all of the chemical bonds. Okay, we're taking the energy from those chemical bonds and we are using it to power the synthesis of ATP. So for one glucose molecule, we're getting between 36 and 38 ATP. That's really a lot if you think about it. All 6 of the carbons from the initial glucose molecule are turned into carbon dioxide, and all of the electrons and hydrogen and protons, electrons and protons making hydrogen, all of those end up with oxygen to make water. The locations, glycolysis happens in the cytoplasm no matter what. That intermediate step right before the Krebs cycle happens in the cytoplasm. If you are a eukaryote, the Krebs cycle and the electron transport chain happen in your mitochondria. If you're a prokaryote, the Krebs cycle happens in the cytoplasm, and that electron transport chain happens in the actual cell's plasma membrane instead of in mitochondria. All right, here's the big picture of what everything looks like. Okay, you have glycolysis to that intermediate modification step to the Krebs cycle to the electron transport.